Hello, my name is Donovan, and my partner, Navneet Bhaskaran, are presenting our cellular respiration video. So have you ever wondered how your body turns a single bite of food into the energy that lets you move, think, and stay alive? Well, the answer is cellular respiration. The purpose of cellular respiration is to break and convert glucose chemical energy into usable ATP, the main energy currency for all living organisms. It is a very complicated process, so we will break the process down for you. There are two types of cellular respiration: aerobic and anaerobic respiration. Aerobic respiration occurs when oxygen is present, and it is done by plants, animals, and humans. Anaerobic respiration, however, occurs when oxygen is not present. It is done by bacteria, yeast, and muscle cells when there is an insufficient amount of oxygen. Here is a small diagram that we're going to break down later throughout this video.
Now we move on to phosphorylation. Phosphorylation is the generation of ATP. ATP can be generated through substrate level phosphorylation or oxidative phosphorylation. Substrate level phosphorylation is a transfer of a phosphate group from a substrate to an ADP molecule to produce ATP. Oxidative phosphorylation is when ATP synthase combines an ADP molecule with a phosphate group using the energy from the E to make ATP. Now for redox reactions. Redox reactions are chemical reactions that involve electron transfers between substances, where one substance loses an electron, when they are oxidized, and another one gains them, they are reduced. The electron carriers in cellular respiration carry electrons from one step to the other. In cellular respiration, they are NAD plus and FAD. When carrying electrons, they carry them in the form of hydrogen, and they are reduced when carrying them, they are reduced to NADH and FADH2. This happens during glycolysis, pyruvate oxidation, and the citric acid cycle. And then in the ETC and fermentation, they are oxidized back because they are donating their electrons. If you look here at this diagram, you can see this is a very oversimplified diagram, but you can see how the glucose is becoming oxidized into CO2 and the oxygen is becoming reduced into H2O. Now you want to know if the first step of cellular respiration, which is glycolysis. Glycolysis occurs in the cytoplasm. Its products are per one glucose used is 2 net ATP, which means they spend 2 and earn 4, they 2 NADH and 2 pyruvate. Now, glycolysis is susceptible to feedback inhibition when products such as ATP accumulate and bind to inhibit the activity of enzymes like phosphofructokinase, PFK, which is the enzyme that allows glycolysis to start. It can either extremely slow down or completely stop glycolysis from occurring. Now, let's take a look at this diagram. You can see how the glucose molecule is entering the enzyme, and you can look up here to see the input for the reaction. You can see how the NAD plus and the 2 ATP needed for the reaction are being put into the reaction, and you can see after the glucose molecule goes into the enzyme and gets broken down using the 2 ATP, out comes 2 pyruvate, 2 NADH, and 4 ATP, which perfectly represents our earlier product stated. Now we move on to pyruvate oxidation. Pyruvate oxidation occurs in the mitochondrial matrix. Its products are 2-acetyl-CoA, CO2, 2-NADH, and 2-NADH. The purpose of pyruvate oxidation is to harvest electrons from pyruvate and is the preparatory step for the Krebs cycle. Now, if you take a look at this diagram, you can see the 2 pyruvate. They are being taken into the mitochondrial matrix. They are attached to the PDC, which is our enzyme in this case. And if you look up here, you can see the 2 NAD pluses going into the reaction. And if you look down here, you can see the 2-acetyl-CoA after the reaction from the pyruvates, the 2 CO2 that exit the reaction, and 2 NADH. The next step is the Krebs cycle. The Krebs cycle occurs in the mitochondrial matrix, also with the pyruvate oxidation. Its products per one glucose used in glycolysis is 6 NADH, 2 FAD, 2 FADH2, 2 ATP, and 4 CO2. The purpose of the Krebs cycle is to harvest all remaining electrons, release carbon dioxide, and generate as many electron carriers as they can. Now, if you take a look at this diagram, you can see the 2-acetyl-CoA entering the Krebs cycle, 2 ADP entering the Krebs cycle, and 6 NADH and 2 FAD and 2 FA 2 6 NAD plus and 2 FAD plus entering the Krebs cycle. As we follow along the Krebs cycle, you can see how all the carbon is removed, all the electrons are trapped in the carriers, making 6 NADH and 2 FADH2, and at the very end, you can see the two exit. Now the next step, it occurs in the inner mitochondrial membrane. It takes the electrons from the NADH, the F8H2, and it puts them through an electron transport cycle. If you take a look at the diagram, you can see this. You can see how the electrons enter the ETC, and the electrons pump protons out of the mitochondria into the intermembrane space. This creates a proton gradient. The hydrogens with this proton gradient will then flow back into the cell through ATP synthase. This motion of going through ATP synthase makes ATP. Now, we will come back to the protons in a sec. After the electrons are very low energy ETC, are very low energy electrons. They just spent all their energy pumping all these protons out of the cell to create this proton gradient, and now they're very low energy. Not many things want low energy electrons, but oxygen does. So oxygen will take the electrons and take the protons that were just pumped back into the cell and combine them to make water, and that water will then be released. And that leads us to our products, which is 34 net ATP, 6H2O. And the overall purpose of the ETC is to make ATP by using the proton gradient and ATP synthase.
So now we're going to be talking about lactic acid and alcoholic fermentation. So both types of fermentation occur in the cytoplasm, and their purpose is to regenerate NAD+, which is an empty electron carrier in the oxidized form, and allow glycolysis to continue and generate small amounts of ATP in the absence of oxygen. So the products of alcoholic fermentation, which we'll explain more in the next slide, is 2-ethanol, CO2, and 2-NAD+, while the products for lactic acid fermentation is 2 lactates, aka lactic acid, and 2-NAD+. So in alcoholic fermentation, 2 pyruvates attach to the enzyme, 2-NADH go in, and 2-NAD+ is the outcome of the reaction, and as well as 2-ethanol and 2-CO2. It's not shown in the diagram, but it occurs. And in lactic acid fermentation, a similar process, 2 pyruvates attach to the enzyme, NADH goes in, NAD plus, 2-NAD plus goes out, and instead lactates, aka lactic acids, are produced, and they're the products of this type of fermentation.
What is a drug or disease that can affect the steps of cellular respiration? The answer is cyanide. Cyanide prevents the cells from using oxygen, which leads to rapid emergency and fatal outcomes. It is used in warfare, genocide, homicide, and many other infamous cases. The way cyanide works is it binds to a crucial enzyme in the ETC called cytochrome C oxidase. That prevents oxygen from being used and causes a buildup of lactic acid and prevents cells from generating their necessary energy. And now for a bonus video.
So we're going to do a home yeast fermentation lab. So right here we have 3 different bottles. This one is a control variable. It has 1 teaspoon of yeast and no sugar. This has 1 teaspoon of yeast and 1 teaspoon of sugar. This has 1 teaspoon of yeast and 2 teaspoons of sugar. Now we're going to put water. So now I have poured hot water, same amount in each of the balloons, and I've placed balloons on top. And now we're going to time lapse and see what occurs in the next 30 to 1 hour. Unfortunately, I wasn't able to get the time lapse of the process, but here are the final results. So the one with the most sugar concentration has the most inflated balloon, and in contrast, the less inflated balloon has less sugar concentration because there's nothing the yeast to react to. And you can see the fermentation on top, the bubbles. So to explain this, yeast goes through alcoholic fermentation, a type of anaerobic respiration, which takes 2 pyruvates and converts it to 2-ethanol, and that's how CO2 is released actually. So you see CO2 is what inflates these balloons. So a quick fun lab to teach us more about real life applications of cellular respiration in many different scenarios, and in this one we just chose yeast. Thank you very much.
Okay. Thank you for listening.